Hello, this is Gina Piscatelli with a lecture for Anatomy and Physiology 2 on spermatogenesis and how it is hormonally regulated. So first let's talk about kind of just generally the overview of the process of spermatogenesis. It begins at puberty and continues throughout adult life. In adults, adult males, 90 to 400 million sperm are made every single day. And as you have already heard from me, it occurs in the stratified epithelium that lines those seminiferous tubules of the testes. For spermatogenesis to occur, testosterone is required. And there's three different kinds of processes involved in spermatogenesis. The first one is differentiation, and that means differentiation of germline cells. And the second process is meiosis, and that's going to be a cell division process. And then lastly is spermiogenesis, which is really this maturation process that I've spoken of that sperm need to undergo before they're completely functional. So let's start off by reviewing the cells that are located in the seminiferous tubules of the testes. Within the wall of the seminiferous tubule, there's two kinds of cells: spermatogenic cells and these cystenocytes, which I always call Sertoli cells. The spermatogenic cells might be in various stages. They might be the completely undifferentiated germline cells that have 46 chromosomes. They might be primary spermatocytes that have 46 chromosomes, or secondary spermatocytes that only have 23 chromosomes. And then finally, there's spermatotids that have 23 chromosomes, and they're developing a flagellum while their head is still adhered to the epithelium. So two kinds of cells: spermatogenic cells at various stages, and these Sertoli cells, also called cystenocytes. In addition, outside tubules are interstitial cells, or cells of Leydig. And so the cells of Leydig are located in this little triangular region in both pictures. In the lumen, you'll find mature sperm, and you can see their flagella here. So now let's talk about the functions of those cells. You already know all the spermatogenic sites are undergoing spermatogenesis. We're making sperm with them. And the cystenocytes, or Sertoli cells, are supporting this process by making growth factors, which cause growth of cells, making nutrients needed for the whole ATP production that's needed during spermatogenesis. And then finally, those cells make something called androgen binding protein. It does just what it says. It binds up androgen, and the androgen that's prevalent in the testes is testosterone. The cells that make the testosterone are these interstitial cells. So let's talk first about the processes of spermatogenesis that involve these cells going through the various stages. The two processes involved are differentiation and meiosis. I suppose you could say mitosis is involved, but I don't want you to get too confused. So the stem cells that have 46 chromosomes, or you could say they're diploid, 2N, are called spermatogonium, or gonia for plural. So one spermatogonium is a stem cell, and stem cells divide. That's what they do, to replenish other cells in our other organs. But in the testes, what the stem cell does is it divides by the process of mitosis, and the two daughter cells are of different types. They're both diploid because we just did mitosis. So they should be identical to the parent spermatogonium genetically. One of the daughter cells is just going to stay like its parent, a stem cell, in order to replenish the number of stem cells. Because once a spermatogonium divides, it's not there anymore. So one of the daughter cells assumes its role. The other daughter cell is what's destined to become sperm eventually. So this daughter cell, we could call it a spermatogonium, I don't know what else it's called, differentiates. That means certain genes turn on. And when it differentiates, it becomes a primary spermatocyte. It is still diploid. It still has 46 chromosomes. This is the starting cell for meiosis. So meiosis 1 is the first division that takes place after the DNA has been copied. So first the DNA is copied in the primary spermatocyte, then meiosis 1 takes place. During meiosis 1, the homologous chromosomes are separated from each other, so that the two daughter cells that form are haploid, and each one contains just 23 chromosomes unpaired. These daughter cells are called secondary spermatocytes, and those two daughter cells will undergo the second division of meiosis, called meiosis 2, and during meiosis 2, sister chromatids separate. The products of meiosis 2 are called spermatotids, and they are also considered haploid. So far we've talked about two processes: the differentiation of spermatogonia and then meiosis. We started with a spermatogonium and made primary spermatocytes by differentiation. Then we underwent meiosis to eventually, after the second meiotic division, make spermatids. These are still not functional, and they're still embedded in the wall of the seminiferous tubule. These spermatids still have to mature into functional sperm, and that involves another process here, and we're going to talk about that in a little bit. So I'm going to review a little bit of kind of anatomy of meiosis. It's kind of interesting. So this is the outer wall of the seminiferous tubule up at the top. And down here is the lumen. And I guess you can tell that because there's some mature sperm floating there. So meiosis actually starts at the periphery of the wall of the seminiferous tubule. So here's where we have spermatogonia. and then differentiation occurs and you get primary spermatocytes. One of those is going to stay a germline cell or a spermatogonium. The other one is going to go through meiosis and become a secondary spermatocyte. Now notice what we're doing as we go through these processes. We're headed towards the lumen. So the process of meiosis from a primary spermatocyte is going to occur towards the lumen of the seminiferous tubule. So here we have secondary spermatocytes, and then closer to the lumen we have the spermatids. And they are still not mature, you can tell, because they don't even have a flagellum. Now, how do they move? Well, these Sertoli cells or sustenatites are yellow in this. That's their cytoplasm. And here's our nucleus. It's kind of blue. They have tight junctions in between them, the different Sertoli cells. And these junctions open up to allow the movement of the developing spermatocytes. It's kind of interesting. It also forms a blood-testis barrier, but we're not going to talk about that. So these spermatids that form are non-motile. They can't move other than just by the proliferation of cells at the periphery kind of pushing them through these tight junctions to get towards the lumen. In this picture, you see a process occurring. These four products of meiosis II are kind of attached to each other. And they're going to separate, and maturation is going to take place. But right now, they're still considered non-motile. They're just developing into mature sperm by a process called spermiogenesis. Now, the sperm that are produced by spermiogenesis, these mature sperm, they do not use their flagellum to move. Even though they have the capability, they do not use it yet. So they are also still considered non-motile. They don't become fully motile until they enter the female reproductive tract. So the process of spermiogenesis, when spermatids become mature sperm, which are also called spermatozoa, include forming what's known as an acrosomal vesicle at the head, and the acrosomal vesicle will be at the very tip, forming a flagellum, and getting rid of any extra cytoplasm from the secondary spermatocyte. However, all the mitochondria that were in the cytoplasm need to be maintained. And we will see that those mitochondria kind of line up towards the base of the flagellum near the head. And that's going to help make ATP to fuel the kind of circular wave-like motion of this flagellum. So I've drawn a little picture, but this picture is better. In spermiogenesis, we start with this secondary spermatocyte. That's this picture A right here. And the first structure that forms is called an acrosomal vesicle, and it's shown in green in this region. Then the flagellum forms, and that involves centrioles assembling microtubules inside the cell and extending into the flagellum. Now what happens is excess cytoplasm is going to be expelled, But all the mitochondria that are orange in this picture, you can see stay close to the base of that flagellum. So eventually, in a mature sperm that's completely undergone spermiogenesis, has a head with a nucleus and 23 chromosomes in that nucleus, a tip that's called the acrosomal vesicle, which is really important for penetrating the egg, the base of the tail that has the mitochondria, and all of the microtubules in the tail flagellum. So here we see the process of spermatogenesis occurring from the periphery towards the lumen, and these 4 spermatids are undergoing spermiogenesis. And those 4 processes we just talked about what's happening to them. But how do they release from this Sertoli cell that's kind of surrounding them? The Sertoli cell, or the sustenocyte, retracts. At the same time, secretes fluid towards the lumen and towards the cells. So the flow of the fluid into the lumen and throughout the whole duct system of the testes, as well as the vas deferens, creates this pressure because the fluid is continually secreted. And it basically dislodges these mature sperm that we still consider not motile because they don't use their flagellum until they're ejaculated. And even then, they don't use it until they get into the female reproductive tract. This picture gives you an idea, I think, for the movement of fluid. It's a piece of artwork created by a medical illustrationist. And here we have the wall of the seminiferous tubule, and I think this gives you kind of a dynamic view of the way fluid and sperm are kind of being pushed towards the lumen as more and more fluid is secreted. And you even see a spermatocyte that's being released by the cystinocyte cells. Some of those are cystinocyte cells. This whole process is controlled by hormones. So next we're going to look at how hormones control the functions of spermatogenesis, or the processes of spermatogenesis. At puberty, there's several areas in the developing brain, because if you recall from psychology, the brain isn't completely developed till at least age 26. But there's several areas in the brain that begin signaling the hypothalamus. And in response, the hypothalamus makes gonadotropin-releasing hormone, GnRH. And here's the hypothalamus region. This hormone, gonadotropin-releasing hormone, travels in the hypophyseal portal system to the anterior pituitary. The anterior pituitary cells respond by making 2 hormones, LH, luteinizing hormone, and FSH, follicle stimulating hormone. These are also called gonadotropins, or they're categorized as gonadotropins because they have a tropic effect on the gonad. And a tropic effect means growth or stimulation of. So both of these are going to act on the testes. It turns out that luteinizing hormone stimulates the testes to release testosterone, another hormone. And follicle-stimulating hormone stimulates the testes to release androgen-binding protein. And you may remember that there's different cells in the testes that make testosterone, then make androgen-binding protein. So two different kinds of cells in the testes. So if we look at it more schematically, we see that the hypothalamus makes gonadotropin-releasing hormone. That causes the anterior pituitary to make two gonadotropins, FSH and LH. And those both act on the testis, or testes for plural. Follicle-stimulating hormone causes the Sertoli cells to make androgen-binding protein. Luteinizing hormone causes the interstitial cells to make testosterone. Now, testosterone has a negative feedback effect on both the hypothalamus and the anterior pituitary. The negative feedback then would inhibit secretion of GnRH and these two gonadotropins, and that would then lower eventually the level of testosterone as well as androgen binding protein. There is another hormone involved called inhibin. And inhibin, which is also made by the testicle, actually only affects FSH. So inhibin is going to act only on the pituitary to decrease FSH. That means that when testosterone levels are high or normal, inhibin, mainly high, inhibin will prevent that testosterone from staying in the testicle, because what inhibin does is it lowers FSH secretion, which lowers androgen binding protein, and so then less testosterone is kept in the testicle. And we're going to review why that's important in a second. First, let's just review why androgen binding protein itself is important and the cells that make each of these. So this is a picture that I've used many times of a seminiferous tubule, and there are cells in the wall trying to undergo spermatogenesis. In order to do so, these spermatogenic cells have to have testosterone around. This testosterone, though, is not made within the seminiferous tubules. It's made out here by the interstitial cells, which diffuses away and might even get into the bloodstream here, because I see a blood vessel. So to keep testosterone levels high enough, the Sertoli cells secrete this protein that binds up the testosterone and keeps it local, so that there's enough testosterone kept within the testicle to permit these spermatogenic cells to undergo the division process, spermatogenesis. So overall, we know that gonadotropin-releasing hormone causes the anterior pituitary to release LH and FSH. LH stimulates the testes to secrete testosterone. FSH causes the Sertoli cells to make androgen binding protein. That promotes spermatogenesis. The presence of this testosterone activates it. But testosterone is needed elsewhere. It's needed to maintain all the cells in the ducts and glands and the penis of the whole male reproductive tract. It's also needed in other parts of the body to form these secondary sex characteristics, like the growth of hair on the face, that kind of thing. And overall, testosterone boosts the basal metabolic rate, and it must act on the brain somewhat, as well as glands, because it's involved with sex drive. So let's say spermatogenesis has occurred enough, right? In that case, you don't really need the androgen binding protein anymore, but you still need the testosterone. The testosterone still has to be made. It just doesn't have to be kept in the testes. It needs to go elsewhere. So there's another regulatory hormone that I mentioned previously called inhibin, made by the testes. And all that it does is inhibit FSH secretion by the anterior pituitary. That means that androgen binding protein won't be made anymore. This allows testosterone secretion to continue because LH is still affecting the Leydig cells of the testes to make testosterone. So we've got lots of testosterone around, but not a lot of androgen binding protein. So this inhibits spermatogenesis, but it doesn't affect testosterone's other actions. That's what inhibin is all about. Okay, that is the end of the male reproductive system. Our last topic will be the female reproductive system, and that will be in the next lecture. Thank you very much for listening.